Welcome to our lecture on hypothalamic nuclei and functions. Imagine you are the CEO of a massive, complex factory that runs 24 hours a day, 7 days a week. You cannot possibly go to every single machine and tell it what to do. Instead, you rely on specialized department managers. In the human body, the hypothalamus acts exactly like this brilliant, multitasking CEO. And today, we are going to meet its managers. Building on our CEO analogy, let us look at how the hypothalamus actually operates. Why does this matter? Because without it, your body would not know when to eat, sleep, or react to danger. Sitting deep inside your brain, the hypothalamus constantly checks internal factory conditions, such as your body temperature, hydration levels, and energy stores. Instead of human managers, it uses clusters of specialized brain cells called nuclei to handle these different tasks. Each nucleus has a specific job, such as telling you when you are hungry or when to release stress hormones. Now that we understand the overall role of the hypothalamus, we need to define what these managers actually are biologically. In neuroanatomy, a nucleus is a distinct cluster of neuronal cell bodies within the central nervous system that share a similar structure and function. Situated in the diencephalon, just above the pituitary gland, these nuclei serve as a crucial neuroendocrine bridge. They translate electrical neural signals from the brain into chemical hormonal signals. Some nuclei synthesize hormones that travel down a special blood vessel system to the anterior pituitary, while others send direct hormones down nerve axons to be stored in the posterior pituitary. Understanding this neuroendocrine bridge leads us to appreciate just how vital these structures are for our survival. The importance of the hypothalamic nuclei cannot be overstated. They are the literal anchors of daily functioning. Without their precise coordination, your body would completely lose its ability to adapt to the outside world. For example, if the temperature-regulating nuclei failed, a simple walk on a hot day could lead to fatal heat stroke because your body would not know it needs to sweat. In clinical medicine, understanding these nuclei is vital for diagnosing complex endocrine disorders, such as central diabetes insipidus, where a specific nucleus fails to produce enough water-retaining hormone. Since we know what happens when these nuclei fail, let us identify the specific department managers and their unique responsibilities. First, we have the paraventricular nucleus, or PVN, and the supraoptic nucleus, or SON. Both are famous for producing oxytocin, the bonding hormone, and antidiuretic hormone, known as ADH, for water retention. The PVN also produces corticotropin-releasing hormone, which triggers your body's stress response cascade. Next is the arcuate nucleus, a master regulator that controls growth, reproduction, and feeding by releasing hormones like gonadotropin-releasing hormone and growth hormone-releasing hormone. Beyond stress and growth, the hypothalamus also manages our daily behaviors and rhythms. We have the ventromedial nucleus, which acts as your satiety center to tell you when you are full. Working in opposition is the lateral hypothalamic area, which acts as your hunger center to drive you to eat. Finally, we have the suprachiasmatic nucleus. This incredible cluster of neurons serves as your body's internal biological clock. It uses light signals from your eyes to regulate your circadian rhythms and sleep cycles, explaining why you naturally feel sleepy when it gets dark. Having discussed the functions of these nuclei, let us now visualize exactly where they are and how they connect to the rest of the body. In this diagram, you are looking at a cross-section of the brain, focusing on the hypothalamus and the pituitary gland just below it. Notice the distinct colored clusters representing the nuclei we just learned about, like the PVN, SON, and arcuate. Observe the two distinct pathways leading downwards. The blood vessel pathway carries releasing hormones to the anterior pituitary, while the neural pathway carries long axons directly into the posterior pituitary to release oxytocin and ADH. You can also see a feedback loop returning from peripheral glands back to the brain, which we will explore shortly. Now that we have seen how hormones are released from these anatomical pathways, we must ask, what happens to them once they enter the blood? While endocrinology is heavily rooted in biology, the way hormones travel and degrade is governed by strict mathematical and physical laws. When a nucleus releases a hormone like ADH, it does not stay in the bloodstream forever. It is continuously cleared by the liver and kidneys. This clearance follows a pattern known as first-order exponential decay. Understanding this decay is crucial for doctors when they need to administer synthetic hormones or calculate how long a natural hormone will remain active in a patient's system. To quantify this clearance process, we rely on a fundamental mathematical model. This formula comes from the natural laws of continuous decay, where the rate of removal is proportional to the amount present. Let us break down the symbols. C of T represents the concentration of the hormone in the blood at any given time t, usually measured in picograms per milliliter. C0 stands for the initial concentration immediately after release. The constant E is the base of the natural logarithm. The variable K is the elimination rate constant, telling us what fraction of the hormone is removed per unit of time. If K increases, perhaps due to better kidney function, the concentration drops much faster, meaning the biological effect is shorter-lived. Before we move on, let us see this formula in action with a concrete clinical scenario. Suppose a patient's supraoptic nucleus releases a burst of ADH. We are given an initial concentration of 12 picograms per milliliter, an elimination rate constant of 0.04 per minute, and we want to find the concentration after 15 minutes. We chose the exponential decay formula because ADH is cleared continuously by the kidneys. First, we substitute our specific values into the equation. We multiply the rate constant by the time to get an exponent of negative 0.6. Evaluating e to the power of negative 0.6 gives us approximately 0.5488, meaning about 55% of the hormone survives. Multiplying this by the initial 12 gives us our final answer. Physically, this tells us the ADH signal is short-acting, allowing the hypothalamus to rapidly adjust to new conditions. We've just calculated how a hormone fades away, but how does the hypothalamus know when to stop releasing it in the first place? This brings us to negative feedback, a critical concept in endocrinology. The hypothalamus regulates itself by listening to the very signals it creates. When a hypothalamic nucleus stimulates a peripheral gland to release a hormone, that peripheral hormone travels back to the brain and acts as a brake on the nucleus. This prevents the overproduction of hormones and maintains perfect homeostasis. For instance, the paraventricular nucleus secretes CRH to trigger cortisol release, and that same cortisol eventually travels back to inhibit the PVN. To understand exactly how strong this braking effect is, we can model it mathematically. This linear formula represents how a peripheral hormone directly suppresses the output of a hypothalamic nucleus. Let us define the terms. S is the actual current secretion rate of the hypothalamic hormone. S0 is the baseline secretion rate, which is how much the nucleus would produce if there were no inhibition at all. The variable X is the concentration of the inhibiting peripheral hormone circulating in the blood. Finally, M is the sensitivity factor, which dictates how strongly the nucleus reacts to the inhibitor. If the sensitivity factor M is large, even a tiny increase in peripheral hormone X will cause a massive drop in the secretion rate S. Now that we have our feedback formula, let us apply it to the paraventricular nucleus, regulating stress. We are given a baseline CRH secretion rate of 50 units per hour, a sensitivity factor of 2, and a current cortisol concentration of 15 micrograms per deciliter. We want to find the new suppressed secretion rate of CRH. We use the linear negative feedback equation because it perfectly models this biological braking system. We substitute our values into the formula. Multiplying the sensitivity factor of 2 by the cortisol level of 15 gives us 30 units of inhibition. We subtract this 30 from the baseline of 50. Our final answer is 20 units per hour. Physically, this proves that because the body has enough cortisol, the PVN automatically dials down its command signal to prevent excess stress. We have covered a tremendous amount of ground today, so let us review the most important ideas. First, remember that the hypothalamic nuclei are specialized clusters of neurons that act as the indispensable command centers of your body. They are the neuroendocrine bridge, translating brain activity into actionable hormonal signals to keep you alive and balanced. Second, we learned that these hormonal signals are temporary. Their clearance from the bloodstream follows strict mathematical rules of exponential decay. Finally, these nuclei are tightly controlled by negative feedback loops, ensuring your body never overreacts. By mastering these concepts, you now possess the foundational blueprint for understanding all complex endocrine pathways. Excellent work today.